Greetings, everybody. Welcome to our Chapter 9 lecture on population distribution and abundance. Populations are groups of individuals of the same species that interact with each other because they're living in the same space over the same time period. So, populations, the important part of this definition is that they're the same species. They interbreed with each other. And there are two basic categories of studies that ecologists do relating to populations. Those asking about the distribution of populations, and those examining what we call population abundance. And ecologists try to understand what factors determine both the distribution and abundance of different species. When studying populations, it's not always easy to tell what a single individual organism is. And an example I can give you are aspen trees that form huge numbers of clones that are all genetically identical to each other. And the largest aspen grove is also the largest living organism on Earth. And since it's all considered to be the same genetic individual, even though when you look at it, it looks like 47,000 different trees over 107 acres. Instead, it's considered one genetic organism. And it's thousands of years old, and unfortunately, it's declining, and it's believed to be due to climate change, very rapid climate change. Here's an example of an aspen grove showing you how it looks like different trees, but in fact, these all might be considered the same individual because they may all be clones of each other that have reproduced asexually. Ecologists seeking to quantify populations report populations as either the absolute population size, which is the number of individuals in the population, or more commonly population density, which is the number of individuals per unit area. That gives you more information than just the number of individuals. As an example, let's say we have a 20-hectare island and we have 2,500 lizards. The population size would be 2,500. That would be something that we could count potentially or estimate according to some methods that we're going to talk about later during this lecture. And the population density would therefore be 125 per hectare. Okay. Now, as a general rule, there is an inverse relationship between population density and body size, meaning that population density is going to be lower for larger organisms, and it's going to be the largest for very small organisms, something like mice. So here's a graph showing you that as the number of individuals per kilometer squared goes down, body mass goes up. So, for example, you would have fewer elephants per square kilometer than you might have whatever this cute little rodent is. Now, distribution and abundance depends directly on the stability of the habitat to the organism in question. Some species require very, very specific habitat conditions in terms of abiotic features, like moisture, temperature, pH, which relate to sunlight, could be characteristics of the soil, like what nutrients and at what level the nutrients are at. And so some species are very specific. And some are more flexible and can survive over a broader range of characteristics. So this is also something that population ecologists investigate. And an example of two species which have different habitat suitability tolerances It relates to creosote and the saguaro cactus. And creosote is an example of a species that is actually very tolerant of both dry and cold conditions. So it occurs fairly widely across North American deserts. Here you can see a picture of creosote. The saguaro cactus can tolerate dry conditions fairly well, but it's very sensitive to cold temperatures. So its distribution is going to be much more limited in where it can survive compared to creosote. Does that make sense? I'll show you a map of the two of them together. Let's see if you can decide which is which. It might help if I point out to you this boundary in Arizona, south of which the temperature never remains below freezing for more than 36 hours. You'll notice that below that line is where we find which species. Saguaro. And you can see that creosote is much more widely distributed. Right. So species, they in addition to abiotic factors determining the distribution and abundance of populations of species, biotic factors can also determine where populations can occur. Remember, biotic means living. So biotic interactions can include things like herbivores, predators, competitors, parasites, pathogens. Specifically in Australia, there's an example of a species that's a good example of a pest species that was actually introduced to the island and quickly became a pest, taking over yards and roads, and it took over a whole bunch of natural areas at the expense of other species that are native to Australia. And so, in order to control the populations of this invasive cactus, Opuntia, the moth Cactoblastus was released. It's a great name, Cactoblastus. And it feeds on the cactus, so it could be used for what we call biological control. Biological control means controlling the populations with another biological organism, as opposed to something like a chemical. So here's a picture of some plots showing you Opuntia in its abundance. And then after Cactoblastus was released, here are those same plots. You can see they've been decimated. So this is an example of a biotic control of population um, uh, dispersal. Well, not dispersal, population existence. Now we'll talk about dispersal. So what is dispersal? Dispersal is the ability of individuals to move from one area to another. And even plants have the ability to disperse, even though animals typically can disperse much more greatly. So species vary tremendously in their abilities to disperse. Even two plants can be very different from each other and have different dispersal strategies. And so trees with large, heavy seeds, they may depend on a mammal or some other organism to eat and disperse those seeds. A smaller plant like this dandelion may use the wind to spread seeds or gametes. So plants have different strategies for dispersal. And sometimes there can be significant barriers, ecological or other barriers to dispersal, like just simply the presence of a desert or an ocean or habitat that just happens to be very unsuitable for the species of interest. Or it could be at something at a much smaller scale. It could be something like a sidewalk or a road or a power line being cut through a forest. All of those things can be a barrier depending on the size and ecology of the species. So habitat fragmentation is the process of creating these barriers that prevent natural species dispersal. And next to habitat loss, habitat fragmentation is the greatest threat to species diversity and to species loss today. Humans have clear-cut much of existing habitat. So even if it still remains, it exists in small isolated patches. And to combat some of those negative effects of fragmentation, here is an example of what is called a corridor, a habitat corridor, made to alleviate the barrier that this road has created in this forest. This is out west. These types of bridges, overpasses, or underpasses even have been made with a lot of success to reconnect previously isolated or previously fragmented habitats and allowing species now to move safely between them. So what does the term geographic range mean? The geographic range includes all areas and all habitats that are occupied during all the life stages of the organism in question. So this is the monarch butterfly, which we've talked about already in this course. And the geographic range of the monarch butterfly, is it large or small? It's a large range because they migrate far distances. And migrating, I wanted to go ahead and define specifically to you. It's moving from one area of a suitable habitat seasonally, usually to track temperatures and resources. And for the monarch, what stimulates it to migrate south in the wintertime to Mexico is the shorter days and the dying off of milkweed and colder temperatures. So some species have very long migrations where they spend time in two separate habitats that are very far away from each other, typically seasonally. So dispersal and dispersion are two different things. We've been talking about dispersal, the movement of individuals from one area to another. Dispersion is the spatial arrangement of individuals within a population, and it can be described as being a regular dispersion pattern, a random pattern, or a clumped pattern. Clumped patterns are the most common. Let's look at a picture of these three types of dispersion patterns. Populations can be clustered, and usually that's due to resource availability. If resources are located in one area, you tend to have species or individuals kind of flock around those resource areas. You can have evenly spaced dispersion, which is far less common. This is a breeding ground, and it looks like these penguins have decided that they've made their nests an exact distance away from each other to be far enough apart, yet to still live as a breeding group. And then you have random dispersion. And random dispersion is just what it sounds like. It could be due to things like windblown dispersal. There's just no real rhyme or reason. It's a random pattern. All right. So as we're studying populations, it's often impossible to count or identify every single member of a population. What we call a complete count of individual organisms, that usually is not able to be done, especially over large areas. So ecologists have to estimate carefully to get an idea of what real population sizes are. And very often, instead of the exact or the absolute population size, we usually refer to the relative population size. For the example, the relative population size is the number of individuals in one time period or place relative to the number in another. So you kind of compare between areas, how many are here versus there. And this might be inferred from scat, which is feces, it might be inferred from things like prints, or from sightings of actual individuals. Also, the effort needed to view or catch individuals can be incorporated into this as well. Ecologists might conduct what are called area-based counts in order to sample a representative area of a larger population. So instead of counting every single coral of a certain species on this reef, which might go on for miles and miles, instead, what they're doing is using what's called a quadrat. And in this example, the ecologist is using a quadrat to count the number of relevant individuals within that square or quadrat. And this is much more feasible than counting an entire reef. It doesn't work well for measuring organisms that can leave the quadrat, so for mobile organisms. So unless you're looking at very, very tiny organisms within a decent-sized quadrat, this is usually for immobile species. And you use a number of different quadrats to sample that are representative of the larger area that you're trying to estimate the population of. So I used this term, a quadrat, and a quadrat is basically just a sampling area of a delineated size. Usually they're 1 meter squared, but obviously depending on what type of organism you're studying, they're going to be different sizes. It can be much smaller than that. So individuals are counted in several quadrats, and counts are averaged to estimate the population size. So it's not that you go and you just measure one quadrat, you measure a number. And very often you'd measure them over time, and you'd measure them randomly to make sure that you're not getting a biased population size estimate. So those quadrats must provide a good representation of the entire area and are chosen at random or placed on a grid. As an alternative to area-based methods, distance-based measures are also sometimes more appropriate for measuring the number of individuals in a population for various reasons. Distance measures are used to determine the distance from a line or a point and can be converted into estimates of abundance. So in a line transect is basically just a straight line that you walk. Typically you lay down a measuring tape to measure your transect as you walk. And in line transects, the observer walks a straight line and measures the distance of any nearby relevant individuals to the line. So if you're studying a specific tree species and you're moving across the screen this way, the direction of travel, according to the arrow, you might come upon distance 1 and see a tree that you're trying to count and estimate the population size this distance from your transect. And then you might walk a little further down and you measure one that's a little bit further out. Bless you. My dog just sneezed. And then in addition to line transects, there are mark, release, recapture studies, or just mark recapture studies. These are used for mobile organisms. This is an example of the process that was shown to you with Monarch Watch, where they tagged the monarch butterflies with stickers and then let them go to continue their migration down to Mexico. That's an example of a mark, release, recapture study. And so these are good for organisms that move long distances. And during this process, the observer is going to capture and mark all possible individuals and make sure that they're marked clearly and carefully, and then release them. And then come back at a later date and do it again. The ratio of marked to unmarked individuals can then be used to estimate population size. So let me give you an example of that. Here's an example with salmon. Stop. You can see the radio tags on the salmon right here. And in this example, let's say we go out one day and we mark and capture 23 salmon. Okay? We're trying to estimate the size of this salmon population. And so we capture and we radio tag, we mark 23 of them. And then later, at a later date, we come back and 15 salmon are captured. 4 of them were marked the first time that we came out. So 4 of the 15 that we catch the second day, had been captured the first day. We use that information, okay? And then N is our total population size. What you can see is if you multiply M, the number during the first mark, times the second recapture, and then divide that number by the number recaptured, you get an estimate of 86 individuals in that population. Okay, so I would be able to do a simple calculation like this to estimate population size. Okay, thanks for talking about populations. We will pick back up talking about how populations grow, looking at the human population in Chapter 10, which is coming up next.